Thank you for the invitation to the Sigmund Freud University. My name is Alexander Haselberger. I'm from University of Vienna, and I would like to discuss with you aspects of a personalized healthcare and salutogenesis. So I would like to go with you through some aspects of what means personal genetics and epigenetics, the gut immune system brain axis, and personalized prevention and intervention. When we are talking about personalized healthcare, we especially I think we need to discuss what is really a person, what's the dispositions, is it really possible to identify personal health hazards, so-called Achilles ferse, what are personal ways for prevention and how to develop strategies for coping in the sense of a salutogenesis. Already Greek philosophers discussed about the influence of nature, nurture. What is a person? Does it come from the environment, or is it born with us? And Socrates and Plato were thinking that it doesn't come so much from the environment. But Aristotle had another theory, and this was the theory that most of a person's personality comes from the environment. And also in the Middle Ages, in the times of Scholastic, Thomas von Aquin's concept based on Aristoteles was, of course, that the image of man is a similar thing like the image of a god. And it's the main idea that it is the soul's possibility to exercise influence on the body. And we started to think about really what is the soul. In the time of ascendance of natural science, in the ages of enlightenment, there were again different ideas. René Descartes was thinking that being self-conscious in the way is integral to having a personal identity. So he was claiming the famous cogito ergo sum. But Francis Bacon believed still that human beings are born in total ignorance, and all the substance of a personality is derived from experience. Modern molecular biology was, of course, a way to the rising of genetics. And from the times of Gregor Mendel, from Watson and Crick and the DNA, to 2003, it was a long way to start the Human Genome Project, which was going from 1990 to 2003. So what was telling us the Human Genome Project? In principle, it was telling us the structure of our DNA, that proteins are coded by just 1 or 2% of the DNA, that the rest of the DNA is retrotransposons, heterochromatin, other sequences. Now we learn that in these non-coding sequences, there are also important regulatory elements like the microRNA. But much more important was, what about the genome of a single person, and where do the differences between persons are coming? And this knowledge is coming from the so-called 1000 Genome Project. This is the sequencing of 1000 genomes of persons of different ethnicity and also different characteristics. So this project explained the variation in the genome, often variations of just a nucleotide position, and one nucleotide may code for a normal protein. Another nucleotide may change of this nucleotide may lead to a low non-functional protein, and the amount of these single nucleotide polymorphisms would contribute a lot to genomic aspects of our diversity and personality. And it's also an aspect of a lot of the genetic analysis done in this area for many aspects of diseases. But there was still a thinking that we cannot explain heritability from these genetic aspects. what is missing to understand the phenotype? It's not so much importance the common genetic variations or the rare genetic variations. We think it's more the knowledge of the environment, gene-environment interactions. And this is really almost the idea of epigenetics. We know, understand epigenetics very well from the twin studies, from the study of monozygotic twins, which have the same DNA sequence, but environmental factors like stress or nutrition change the epigenetic regulation of the genes. And this is responsible that the monozygotic genes having the same genetic equipment can have a very different phenotype and different functions. So our understanding is that the environment, the genetics in their interactions about epigenetics are important for health and for personal aspects. Of course, epigenetic is a central element for understanding of personal developments of complex diseases such as cancers or aging. And we know the so-called hallmarks of aging, the mechanisms which contribute to aging, fast or slow aging, or even the hallmarks of cancer, where we know about the different mechanisms which are typically for the cancerogenesis. We also know about the importance of gut microbiota, and we know that the gut-brain immune endocrine axis is of central importance for personal health, but also for many of our personal characteristics, even psychological characteristics. So the basis for personalization are microbiota, DNA, epigenetics, and environments. But of course, there are phenotypes which are more addressed by genetics, such as height, which is where about 80% of height is correlating with genetics, whereas in the aspect of metabolic diseases, it's about 60 to 70%, which comes from the environment and from the nutrition, which is responsible for these diseases. And this understanding now enables us a preventive personalized healthcare with the use of markers, whereas current healthcare waits for the development of pathologies and the appearance of symptoms before an intervention comes up. The use of markers, molecular markers, can look for possible dangerous developments and can start with interventions already before the appearance of symptoms. So these markers are of central importance. Epigenetic markers, methylation even, can tell us if stress, nutrition can influence us and makes us and presses us in the direction of an accelerated aging, or if you have a slow aging and a healthy aging, that can be shown with epigenetic methylation markers. In the same way, there are epigenetic markers for the development, progression of cancers. And in our lab, we can look for methods of lipid biopsy, where we have markers of cell-free DNA, where we can show if certain central cancers are rising, which type of cancers they are, and maybe we even can look for methods of monitoring and ideas for an appropriate personal intervention in this area. So we are now in the age of a personalized, so-called precision medicine, where we look not so much for the tumor and the tissue, but we look for DNA mutation. And on the other hand, we are going towards a precision nutrition with a specific personal nutritional management and planning and monitoring. So personal health care and salutogenesis are going hand in hand for improvement of personal functions. And this is the need for markers. These markers need to be processed in bioinformatic ways. And this bioinformatic analysis of these markers enables us a personal intervention with exercise, nutrition, supplements, lifestyle, social methods, psychotherapies. And to my thinking, I think it will be always a very specific mix of these interventions for different people. And of course, monitoring of the effects of the intervention, again, using markers. In this area, the spin-off of our department and of my working group, Health Biocare, has set up a system to identify possible dangerous developments, set up a personalized prevention using functional foods, and support also personalized for lifestyle and exercise. And this is now a great tool towards personalized healthcare and personalized nutrition. This all can be done by analysis from a blood spot. This system is very robust and can be stable quite a long time. So it's really a good way towards getting molecular markers for personalized health care. And my personal conclusions and discussions is, well, we are now looking for personalized health care. We know that one size doesn't fit all. We know that we have to treat a single person. We know, or we can think that we go not only in the way of a pathogenesis thinking, but we also need to go to the thinking of a salutogenesis, health promotion towards health, well-being and functioning and coping with problems. So we have identified many of the molecular mechanisms and markers for this, but still we need to think about putting all these things together. So health, is it correlated with lots of these mechanisms, molecular markers, or should we not think, especially at the Sigmund Freud University, that we have to treat not only single aspects of a health, but maybe we should treat the soul of a person. Thank you for your interest and discussion. I would like to meet you again on www.alexander-haselberger.at or www.mypersonalhealth. Stay healthy. Thank you.